Welcome to the Deep Dive. Our mission, as always, is to get you immediately well-informed, and today we are tackling, well, maybe the most defining question of human biology. What is the genetic secret sauce? You know, the thing that gives us complex language.
It's a fundamental question, absolutely. What makes us us in terms of communication?
And for a long time, the scientific community, they thought this was kind of a treasure hunt, right?
Yeah.
Looking for that one single powerful key.
Yeah, the language gene idea.
Exactly. And we all know the star of that period, the FOXP2 gene. That gene was really hailed as the definitive language gene because, well, when it's mutated, it caused these severe generational speech and language disorders.
Right. It gave us a massive insight, no doubt. It showed a clear genetic link to speech mechanics, vocal learning.
Right.
Really important groundwork.
But it also kind of pointed us down maybe too simple a path.
It was a crucial starting point. But yeah, that search for a single master switch, it turned out to be, well, reductive. We know FOXP2 is essential. I mean, the mutation's undeniably linked to developmental disorders, severe ones. Yeah. But it turned out to be nearly useless in explaining the vast normal spectrum of language ability. You know, why are some people naturally just more verbose or poetic or quick-witted, while others might struggle a bit with finding the right word?
FOXP2 didn't hold that key.
It didn't. So the simple single gene model, it didn't work for explaining variation in the general population.
Okay, so if it's not one magic blueprint for a protein, what did researchers realize they had to start hunting for instead? Where did the focus shift?
Well, they realized they had to look bigger, think less about the individual light bulb, and more about the entire electrical grid, the whole operating system, specifically the regulatory layer.
The switches and dials.
Exactly. The focus shifted dramatically from those protein-coding genes like FOXP2 to the vast and honestly still pretty mysterious non-coding regions scattered all across the genome.
The so-called junk DNA that isn't junk at all.
Precisely. These are the genetic conductors, the switches, the volume knobs. They determine when and where those important protein-coding genes like FOXP2 actually get turned on or off.
Okay, so it's about timing and location.
Fundamentally. And the research we are diving into today focuses intensely on these non-coding elements. It introduces a really unique class of genetic innovation, something forged by, well, blistering evolutionary speed.
And these regions have been given a pretty cool name, HAQERs.
That's right. Human Ancestor Quickly Evolved Regions, HAQERs.
So our mission today is to really unpack this. What are HAQERs? How did this rapid evolution happen? And what does it tell us about, well, everything from our brain development, maybe the cognitive capacity of archaic humans like Neanderthals?
And critically, why this rapid evolution might have also introduced some painful vulnerabilities into our genome. There's a potential downside we need to explore.
That is a really ambitious stack of material.
Yeah.
Okay, let's start at the foundation. Who are the people whose DNA actually made this discovery possible? What was the study cohort?
So the core discovery data set, it's rooted in something called the episodic study. It was a large-scale epidemiological investigation done in Iowa.
Capicella.
They initially screened, get this, over 7,200 kindergartners. The goal was originally to identify kids with specific language impairment, SLI.
Wow, 7,200. That's huge.
It is, but from that large pool, the researchers then selected a final, really deeply characterized sample of 350 unrelated individuals, all of European descent.
And the key was deeply characterized.
Yes, and also that these 350 participants were specifically chosen to represent a massive, broad range of language abilities, not just those with impairments, importantly, but also kids with average and even superior language skills.
Ah, so they wanted the whole spectrum.
Exactly, to capture the natural variation across the population. That's crucial if you want to understand the genetics of typical ability, not just disorder.
Got it. And you said deeply phenotyped. I understand that means they went far beyond just a simple test. How extensive were these assessments they used to kind of distill down their core language ability?
Oh, incredibly rigorous. And longitudinal, which is super important. We aren't talking about, a quick vocabulary quiz.
Right.
They used a really extensive battery of language and cognitive measures. They looked at things like oral vocabulary, grammatical completion, how well kids could finish sentences grammatically, the complexity of sentences they could formulate themselves, sentence repetition ability, things like that. And also nonverbal performance IQ.
Okay, a whole suite of tests.
Right.
And longitudinal means over time.
Yes, they tracked these abilities developmentally. They administered the full battery at 3 really critical time points. First, when the children were in kindergarten, then again in 2nd grade, and finally in 4th grade.
Wow, tracking ability over 4 really formative years. That is a massive amount of data per person.
It really is a rich data set.
So how do they condense all those different scores from different tests over different years into something they could actually use for genetic analysis? That sounds statistically complex.
It definitely is. They employed complex statistical analysis, specifically factor analysis. The goal was to distill all those individual scores down into fundamental underlying components or factors. They ended up with 7 factors labeled F1 through F7.
7 factors.
But critically, for the genetic part of the study, they zeroed in on just 3 of those factors, F1, F2, and F3.
Why only those 3?
Because those were the 3 that showed the strongest, most measurable genetic association signal. They carried the most heritability, if you will.
Okay, so F1, F2, F3 are where the genetic actions seem to be. What do those factors actually represent? Are they like grammar and vocabulary?
It's a bit more blended than that. These 3 factors, F1 to F3, they essentially represent a highly correlated mix of oral language skills. So yes, vocabulary and grammar are in there, but also alongside measures of nonverbal IQ.
Interesting. So language skill and nonverbal intelligence are bundled together in these genetic factors.
In these particular factors, yes. The key takeaway is that they weren't measuring just one simple thing. They were measuring the complex, really intertwined abilities that we recognize as, advanced human communication and cognition.
Okay, so we've established why the deep dive into this non-coding DNA, the regulatory stuff, was necessary. The single gene approach, like with FOXP2, it just failed to explain the typical variation we see in language ability across people. Why is it so critical, conceptually, to shift our attention from the gene itself, the protein blueprint, to these surrounding regulatory regions?
Right. It's a fundamental shift in thinking. Think of it this way. The gene, the actual blueprint for a protein, is often very, very old, evolutionarily ancient, and highly conserved across many species.
Meaning the same basic gene exists in mice and chimps and us, doing a similar job.
Exactly. Now, if you want to evolve a complex new trait like sophisticated human language, you usually can't just dramatically alter that fundamental conserved protein blueprint, because that protein probably has like 10 different essential jobs it does throughout the body.
Right. If you mess with it too much, you break other things.
Precisely. That's the core idea of pleiotropic constraints. Pleiotropy means one gene affects multiple traits. So if you change a core gene to improve language, you might accidentally mess up, kidney development or something else vital. Evolution tends to avoid that.
It's like trying to upgrade your car's engine by changing the spark plugs. But those spark plugs also somehow control the brakes and the radio. You'd be hesitant.
That's a great analogy. So the genetic solution evolution often takes is, well, it's clever. Keep the highly conserved protein exactly as it is, doing its essential jobs, but invent new specialized switches, these non-coding elements we're talking about.
Okay.
And these new switches can turn that old conserved gene on only in a very specific place, like, say, the developing cerebral cortex.
The part of the brain crucial for higher thought.
Right. And only at a specific time, perhaps during prenatal development, when the brain's foundations are being laid. This allows for rapid, innovative evolution building new functions without the crippling constraints of breaking those ancient multifunctional systems.
So the innovation is in the control panel, not the core machinery.
That's the consensus now, yes. The human-specific features that really define us cognitively, they seem to be overwhelmingly driven by mutations in these regulatory regions, not so much in the protein-coding genes themselves.
Which means that the abilities wrapped up in those factors, F1, F2, and F3, are core language factors from the EPILI study. They must be built on a staggeringly vast, distributed network of these regulatory elements.
They absolutely are. It's not one switch, it's thousands acting in concert. And the EPILI cohort provided a really interesting clue early on that these factors weren't operating in isolation.
What was that clue?
Well, what the initial crucial findings came from behavioral assessment scores. They used something called the Child Behavior Checklist, the CBCL. Gathered when the kids were in 2nd grade, this was available for about 2/3 of the sample.
Okay, so behavioral data alongside the language data.
Exactly. And they discovered pervasive and statistically significant associations between those core language factors, F1, F2, and F3, and various measures of mental health and neurological stability in the children.
Wow. So even in young kids, the genetic underpinnings for having strong language ability are already intrinsically linked to, well, neurological health and resilience,
Mm.
or the lack thereof.
It certainly suggests that. It hints that the evolutionary process that built our sophisticated language machinery might also somehow carry the blueprints for certain vulnerabilities.
That finding acts as a critical bridge, doesn't it? It sets the stage for later in our discussion, Section 5, where we'll look at the potential evolutionary cost of these language adaptations.
Exactly. It connects the evolution of language ability directly to potential health outcomes. But first, to really study these complex, widespread genetic factors, especially the contribution of specific evolutionary events like the ones that created HAQERs, the researchers needed a new kind of tool, a sharper knife, if you will.
And that's where this innovative ESPGS method comes in.
Precisely. Evolution Stratified Polygenic Score.
Okay, let's unpack this ESPGS. It sounds powerful. But maybe for our listener, we first need to just quickly define a standard polygenic score, a PGS. What is that?
Sure. A standard PGS, a polygenic score, is essentially a sophisticated statistical prediction tool. When you're trying to predict a complex trait, something influenced by many genes like height or risk for heart disease, or in this case, language ability, you don't just look at one gene.
Right, we know that doesn't work well.
Exactly. Instead, you look at thousands, maybe even millions of independent genetic markers, usually SNPs, single nucleotide polymorphisms spread across the entire genome. You figure out the small effect size each of those markers has on the trait based on large population studies. Okay.
Yeah.
Then you add up all those tiny effects for a given individual, weighted by their effect size, to create one single summary score. It's like a genetic risk score or a genetic propensity score. It gives you a prediction, a sort of genetic forecast for that individual's trait.
So a standard PGS predicts your likelihood for a trait based on your overall genetic makeup. But crucially, it can't tell you when in evolutionary history or why that specific genetic contribution evolved, can it?
Exactly, that's the limitation. A standard PGS treats the whole genome as one big pot. That's the key innovation of the ESPGS method developed for this study. It takes that standard polygenic score concept and partitions it. It slices it up based on evolutionary timing or functional annotation.
Slices it up. How does it do that?
It's designed to systematically examine how genetic variants that originated in different evolutionary periods, like, say, variants that arose before the split with Neanderthals versus those that arose after, or variants located in specific functional regions of the genome, contribute to the overall trait prediction.
Okay, so how does it physically, or rather statistically, carve out the genome to test a specific evolutionary moment, like the one that created the HAQERs?
It creates a statistical comparison using two main components. Let's call them the annotation PGS and the background PGS.
Annotation and background. Got it.
So when we want to specifically test the contribution of the HAQER regions to language ability using factors F1, F3, the ESPGS method takes the language trait prediction and jointly models it against two scores simultaneously. First, the background PGS, which is calculated using all the relevant genetic variants outside the HAQER regions, basically, the rest of the genome's contribution.
Okay, a baseline prediction.
Right. And second, the annotation PGS, which is calculated only from the genetic variants found within those specific HAQER regions.
I see. So you have a score for HE only and a score for everything else.
Exactly. And by running the full statistical model, which includes both the background PGS and the HAQER annotation PGS, and comparing its predictive power to a reduced model that only has the background PGS,
you can definitively isolate and measure the unique additional contribution of that specific evolutionary layer, the HAQERs themselves.
that is the crucial step. It moves beyond just seeing a correlation or a statistical coincidence. The ESPGS technique is essentially asking a very specific question. Does this small, rapidly evolved slice of the genome, the HAQERs, predict language abilities significantly better than we would expect, just based on the predictive power of the remaining 99-point-whatever% of the genome?
That's clever. It lets you directly test if an ancient evolutionary burst has a specific measurable impact on a modern human phenotype, like language skill, over and above the general genetic background.
Precisely. It's a powerful tool for linking specific evolutionary events to current human traits and variation.
Okay, so now we have the tool, the ESPGS. Let's apply it to the discovery itself, HAQERs. Human Ancestor Quickly Evolved Regions. We need to really get into the details of how the researchers actually found these 1,581 patches of what sounds like genomic lightning. What was the absolute quantitative standard they used to define them as quickly evolved? It couldn't just be a feeling.
No, definitely not. It was very quantitative. The defining criterion was the sheer density of evolutionary changes, or divergence, when comparing the modern human sequence to the reconstructed sequence of the human-chimpanzee ancestor.
Okay, density of change.
Yes. They weren't looking for slow, gradual change over millions of years. They were specifically hunting for genomic areas that had essentially gone rogue, evolutionarily speaking, since our lineage split from the chimp lineage.
How do they measure that going rogue?
They scanned the genome in small moving windows of 500 base pairs each, and a specific 500 base pair window qualified as a HAQER only if it showed an incredibly high number of evolutionary operations. That means substitutions, one DNA letter changing to another, insertions, DNA being added, or deletions, DNA being removed, separating the modern human sequence from that inferred ancestral sequence.
And how high was that threshold? What number of changes made it quick?
It was set extremely high, designed to capture only the most extreme outliers of evolutionary speed. A region had to exhibit 29 or more of these evolutionary operations within that 500 base pair window.
29 changes in just 500 letters. That sounds like a lot.
It is. Statistically, it represents a divergence density with a p-value less than 10 to the minus 6. That means the chances of seeing that much change happening just randomly by neutral drift were astronomically low, less than 1 in a million.
Wow. Okay, so it's a very stringent definition. And applying that across the whole genome yielded.
It yielded 1,581 distinct HAQER regions. Collectively, they span about 1.41 megabases of the human genome. So they're relatively rare hotspots, not huge chunks, but they represent these concentrated zones of extremely rapid evolution.
Now, what's really fascinating here, and you touched on this, is the distinction they drew between these new HAQERs and the previously defined standard for human evolutionary novelty, the HAQERs, human accelerated regions. Why are these two classes of rapid evolution so distinct? They sound similar.
They sound similar, but the discovery method, and therefore what they represent, is quite different. HAQERs were traditionally identified using a two-step filter. First, researchers looked for regions that were highly conserved across mammals' genomic elements that had stayed pretty much the same for tens or even hundreds of millions of years. Stable, important stuff.
Okay, find the old stable parts first.
Right. Then, within those conserved regions, they looked for places where the human lineage showed an unexpected burst of changes, breaking that ancient conservation. So HAQERs are often seen as evolutionary tweaks or modifications to pre-existing ancient functional elements.
Modifying something old.
Exactly. HAQERs, in contrast, were identified purely based on that raw speed of divergence from the human-chimp ancestor. They completely skipped that initial conservation filter. They didn't care if the region was old and stable in other mammals or not. They only cared. Did it change incredibly fast on the human lineage?
Ah, so HAQERs could be arising from anywhere, maybe even from previously non-functional neutral DNA. Less like fixing up an old house and more like building something entirely new on a previously empty plot of land.
That's a perfect analogy. They are potentially innovations forged de novo from scratch, evolutionarily speaking. And this distinction is really critical because when they checked for overlap, out of the 1,581 HAQERs they identified, only a tiny number, just 6, showed any overlap with the expanded list of known pairs.
Only 6. That's practically no overlap at all.
Exactly. It strongly confirms that HAQERs represent a distinct, previously largely uncharacterized class of human-specific functional evolution. These are regions likely less constrained by ancient functions, allowing for perhaps more radical innovation.
Okay, so we have these incredibly fast-evolving regions. When you see that kind of rapid divergence, there are usually two main potential explanations, right? Was it just a random accident, or maybe because that specific area of the genome just happened to have a higher mutation rate? Or was it driven by purposeful, directional, selective pressure? Evolution favoring those changes?
That's the key question, mechanism. And the researchers found evidence for what they called the dual forces of evolution. It seems both factors played a role in shaping HAQER.
Okay, so a mix of chance and purpose.
Kind of. First, the chance or predisposition factor. They found that HAQERs were statistically enriched, meaning they were located more often than expected by chance, near genomic features known to promote higher local mutation rates.
Like what kind of features?
Things like meiotic recombination hotspots, areas where DNA swaps pieces during sperm and egg formation, which can be mutagenic. Also, regions near the ends of chromosomes, the telomeres and subtelomeres, tend to have higher mutation rates. So HAQERs were often located in parts of the genome that were already, shall we say, mutation-prone.
So they were physically poised for rapid change. They were already kind of living in the fast lane, genetically speaking, just waiting for the evolutionary gas pedal to be pressed.
That's a good way to think about it. And the evidence suggests the gas pedal was pressed hard, and that pedal was directional positive selection.
Ah, the purposeful part. Not random changes, but changes that provided a benefit.
Exactly. Positive selection is the non-random force. It's when new genetic variants emerge that provide some kind of biological advantage, making the individuals carrying them more likely to survive and reproduce. These advantageous variants then tend to sweep rapidly through the population and become common or even fixed, reaching 100% frequency.
How do they detect this positive selection specifically in HAQERs?
They used sophisticated population genetics analyses, looking at the patterns of genetic variation within HAQERs in modern human populations, particularly focusing on African populations, which retain the deepest ancestral human diversity. They analyzed something called the derived allele frequency spectra, or DTOFS.
Derived allele frequency, meaning how common the new version of the DNA letter is compared to the ancestral version.
Precisely. And HAQERs showed extremely strong statistical signatures consistent with positive selection acting on them. The patterns indicated that the variants within HAQERs were much more likely to have risen to high frequencies than expected under neutral evolution.
So the changes weren't just happening, they were being actively favored.
Yes. They even estimated the strength of this selection. The mean selection coefficient says a measure of the fitness advantage for bases within HAQERs was estimated to be somewhere between S.000000635 and S equals 0.0000825.
Is that considered strong selection?
In human evolutionary terms, yes, that's quite substantial for selection acting across many sites. It indicates a powerful and consistent adaptive benefit was driving these rapid changes. These weren't just random noise, they were functional upgrades that helped define the evolving human lineage.
This brings us then to perhaps the most critical piece of this puzzle, the timeline. When did this intense burst of HAQER evolution, driven by positive selection, actually occur?
This is one of the most striking findings. The analyses of sequence divergence patterns, comparing human, Neanderthal, Denisovan, and chimpanzee genomes, pinpointed the timing with remarkable clarity. This rapid divergence, the main burst of positive selection shaping the HAQERs, occurred in an episodic burst primarily prior to the split of the lineage leading to modern humans from the lineage leading to Neanderthals and Denisovans.
Wow, okay, wait, so before anatomically modern humans even existed, and before our ancestors diverged from the ancestors of Neanderthals and Denisovans, that's when these specialized, potentially language-related elements were being rapidly forged by intense selection.
That's what the data strongly indicates. We're talking about innovation happening deep in the shared ancestry of the hominin line, maybe somewhere between, say, 500,000 and perhaps a couple of million years ago, after the split from chimps, but before the later hominin divergences.
That's much earlier than I think many people would assume for complex language-related genetics.
It is potentially quite early, and the follow-up finding is equally important. Maybe even more so for understanding their function later on. After this initial burst of rapid evolution driven by positive selection,
Yes.
HAQERs then transitioned into a state of strong evolutionary constraint within the hominin lineage.
Constraint, meaning they stopped changing so rapidly.
Exactly. Once those key adaptive changes were established in the common ancestor of humans, Neanderthals and Denisovans, subsequent evolution acted primarily to preserve the HAQER sequences. They became highly conserved among hominins. Modern humans, Neanderthals and Denisovans, share remarkably similar sequences in these HAQER regions.
So the upgrade happened early, and it was so vital that evolution then put the brakes on, protecting those regions from further significant changes.
That's the interpretation. The functional innovation enabled by the HAQER changes was likely so important for hominin biology that once it was fixed or nearly fixed in our common ancestor, it became selectively disadvantageous to mess with it further. This suggests the core capacity these HAQERs contribute to was established very early in hominin evolution and was likely shared among these groups.
Okay, so let's recap. We have these 1,581 specific regions, HAQERs, that underwent this incredibly fast, positively selected evolution, mostly before the split between the human and Neanderthal Denisovan lineages. And then they became stable, constrained. The next huge challenge is figuring out what they actually do. Given our earlier conversation about avoiding pleiotropic constraints and the ideas of specialized switches, where in the human body would we logically expect these elements to be active?
Right, function is key. And
Yeah.
given the context of language evolution, the brain is obviously the prime suspect. But it's not just where, it's when. The timing turns out to be absolutely crucial.
So what did the functional analyses show?
Well, initial analyses looking at nearby genes showed some enrichment for things related to olfaction, which is interesting, and also cell recognition genes. More importantly, they looked at 3D chromatin contexts, basically, which genes these HAQER regions physically loop over and touch in the nucleus, suggesting regulatory interaction.
And who are they touching?
They found significant enrichment for HAQERs, making these 3D contexts with genes known to be involved in nervous system development and function. So the brain connection was definitely there.
Okay, connecting to nervous system genes, that makes sense.
But here's the critical distinction that emerged when they looked not just at which genes, but when and where the HAQERs themselves seemed to be active regulatory elements. They compared HAQER locations to maps of known enhancers and regulatory regions from various tissues and developmental stages.
And what was that critical finding regarding timing?
They found that HAQER regions showed essentially no significant enrichment for matching regulatory elements active in the adult brain.
None. So they're not regulating ongoing brain function in adults.
Not primarily, it seems. Instead, the strongest signal, by far, was that their influence occurs predominantly and powerfully during prenatal neurodevelopment.
Ah, so they're involved in building the brain, not running the finished product.
That's exactly the implication. The genomic innovations associated with HAQERs appear to be fundamentally structural. They seem to contribute to the unique wiring, the cellular makeup, the very architecture of the human brain as it is being constructed in the womb.
That's profound.
Yeah.
They're part of the construction crew, setting up the unique hominin brain plan.
That seems to be the picture emerging. Now, let's revisit that idea we discussed, HAQERs being specialized, maybe arising de novo from previously neutral DNA to bypass those pleiotropic constraints. How does this idea translate into their physical regulatory mechanism? What do they look like at the chromatin level?
Right, the actual chemistry of how they work as switches.
This is where the chromatin state analysis gets really fascinating. They found that HAQERs are strongly and significantly enriched for carrying what are called bivalent chromatin marks, or being in bivalent chromatin states.
Bivalent chromatin. Okay, let's break that down. What does bivalent mean in this context? Sounds like two values.
It does, and that's basically it. Imagine a gene's regulatory region. It can have chemical marks, epigenetic marks, that signal activate this gene, like a green light. And it can have marks that signal repress this gene, like a red light.
Okay, on or off?
Bivalent chromatin means that the regulatory element carries chemical marks for both activation, like H3K4 trimethylation, and repression, like H3K27 trimethylation, simultaneously. It's like having both the green light and the red light on at the same time.
So, it's paused, prepared but waiting.
Exactly. It's considered a state of high preparedness, but it's paused or poised. It signifies a regulatory element that's ready to spring into action, but it requires a very specific signal to resolve the bivalency, usually tipping it towards activation.
So it implies a high degree of control and specificity. It's not just generally on or off. It's waiting for a precise cue.
Precisely. Bivalent chromatin is often associated with developmental genes and regulatory elements that need to be activated only at a specific moment or in a specific cell lineage, or perhaps only in response to a very particular environmental or developmental signal. It suggests a high degree of regulatory specificity.
And this fits perfectly with the idea of HAQERs being newly evolved, specialized switches, right? Designed to turn on genes in very specific contexts to avoid messing up other functions.
It fits beautifully. This kind of poised, highly specific regulation is exactly what you might predict for de novo functional elements that evolved to circumvent the pleiotropic constraints associated with modifying older, broadly active regulatory elements. It's also a major point of contrast with many previously identified HAQERs, which are often associated with more broadly active, less specific enhancer states, like those marked only by H3K27 acetylation.
Okay, so the theory holds together beautifully based on chromatin states and location data. But the ultimate proof, the real smoking gun, requires actually putting these specific HAQER sequences into a living, developing system to see if they truly act as enhancers, especially the human hominin version compared to the ancestral version.
Absolutely. You need functional validation. And that brings us to what is arguably one of the most technologically impressive and frankly mind-blowing parts of this whole study, the in vivo functional validation experiments.
This sounds like where the aha moment happens.
It really is. This is where they moved from computational prediction to biological reality. The researchers used a very advanced technique called in vivo single-cell star-seq.
Okay, star-seq. Self-transcribing active regulatory region sequencing. In single cell on top of that, what did they do?
Okay, buckle up. They synthesized DNA sequences corresponding to specific HAQERs. Crucially, they made two versions for each HAQER tested. The modern human sequence, or sometimes the inferred Neanderthal Denisovan sequence, which is very similar, and the inferred ancestral sequence, the sequence present in the human-chimp ancestor before the rapid evolution occurred.
Human version versus ancient version. Got it.
They then cloned these sequences into a special reporter construct. This construct is designed so that if the inserted sequence, the HAQER, acts as an enhancer, it will drive the expression of a reporter gene, allowing them to measure its activity.
Okay, measuring enhancer strength.
But here's the in vivo part. They used a technique called in utero electroporation. They injected these reporter constructs directly into the developing brains of mouse embryos, specifically targeting the cerebral cortex at critical developmental time points, embryonic day 14.5 or 15.5.
Wait, if I heard that correctly. They inserted synthetic DNA representing modern human and ancient human regulatory regions into the developing brain of a living mouse embryo, and then measured which human segments acted as genetic switches inside the mouse brain. That sounds almost like science fiction.
It is remarkably precise and powerful molecular biology. It allows you to test the regulatory potential of a human DNA sequence in the relevant developmental context, the developing mammalian brain.
Okay, mind slightly blown. But what about the single cell part? Why was that important?
That was the other crucial layer. Instead of just measuring the overall enhancer activity in a chunk of brain tissue, they dissociated the electroporated brain cells and performed the star-seq analysis at the single-cell level. This gave them incredibly fine-grained resolution.
Meaning they could see which specific cell types the enhancer was active in?
Exactly. They could map the enhancer activity driven by the HAQER sequences to specific foundational cell types within the developing cerebral cortex. We're talking about the key players. Radial glia, the primary neural stem cells, intermediate progenitors, which amplify neuron production, and newborn excitatory neurons themselves.
And radial glia. Those are incredibly important, right? They're like the master stem cells for the cortex.
They absolutely are. Radial glia are the foundational progenitor cells of the cerebral cortex. They're responsible for generating almost all of the excitatory neurons that populate the different layers of the cortex. Their activity and regulation fundamentally determine the cortex's ultimate size, structure, and complexity. So, if HAQERs are acting as enhancers specifically in radial glia,
they're fundamentally shaping the structure and potentially the size of the human brain right from the earliest stages.
That's the hypothesis, yes. They're influencing the core manufacturing process.
So what was the definitive result? Did the human hominin HAQER sequences actually show enhancer activity? And was it different from the ancestral sequences? This is the key test.
This was the absolute key. They successfully tested 13 different HAQER regions using this complex in vivo single-cell star-seq system, comparing the hominin version side by side with its inferred ancestral ortholog.
And the result?
A clear validation. 6 out of the 13 tested HAQERs showed significantly greater enhancer activity when they used the hominin sequences, human, Neanderthal, or Denisovan. They were often tested together as they are so similar, compared to the activity driven by the corresponding ancestral non-hominin sequence.
6 out of 13 showing a significant gain of function. That's definitive proof, isn't it? It shows the sequence changes that occurred during that rapid evolutionary burst really did create new functional regulatory elements, enhancers, that are specific to the hominin lineage.
It provides the strongest possible functional evidence, yes. It confirms that these rapidly evolved regions are not just sequence curiosities, they are bona fide hominin-specific enhancers active during critical phases of brain development.
Can you give us a specific example, maybe a HAQER that showed this effect and the gene it might be regulating, something tangible listeners can hold on to?
Yes, the example they highlight for HAQER 0059 is a really great illustration, and they even backed it up with traditional enhancer assays using EGFP, a fluorescent protein.
Okay, HAQER 00059.
This specific HAQER is located quite near a family of transcription factor genes called the FOXD4 genes, FOXD4 and FOXD4L. When they put the human version of HAQER 000059 into the reporter system and into the mouse brain, it showed robust, strong enhancer activity driving the expression of the reporter gene, like EGFT making cells glow green.
So the human version worked as a strong switch.
Yes, but when they tested the inferred ancestral sequence for HAQER 0059, it showed almost no significant enhancer activity above the baseline control levels. The switch was basically off in the ancestral version.
Wow, a clear difference. And what do the target genes, the FOXD4 family, do?
Well, intriguingly, the FOXD4 gene family itself has been implicated in fundamental aspects of brain size regulation in humans. Copy number variations, meaning deletions or duplications of the genomic region containing these genes, have been associated with clinical cases of microcephaly, abnormally small brain size, and macrocephaly, abnormally large brain size, respectively.
No way. So this directly links one of these early, rapidly evolved hominin-specific enhancers, HAER 0059, to a genetic pathway, the FOXD4 pathway, that appears to play a role in determining the overall scale and architecture of the developing human brain.
That's the connection, yes. It provides a plausible molecular mechanism. Rapid evolution creates a new enhancer, HAQER 00059, near a key brain size regulating gene, FOXD4, potentially altering its expression during development, contributing to the trajectory of hominin brain evolution. It's a compelling piece of the puzzle.
This research has built a really strong case. The core functional changes in these HAQERs, turning them into hominin-specific enhancers active in prenatal brain development, happened before the split between the lineage leading to modern humans and the lineages leading to our ancient cousins Neanderthals and Denisovans.
That's right. The evolutionary timeline points to these being shared innovations, established early.
Which naturally leads us to a really fascinating, almost haunting question. How did Neanderthals and Denisovans actually score on this HAQR-based genetic scale for language predisposition? If these enhancers were already active in our common ancestor, did they have the same potential?
That is indeed the million-dollar question, maybe the billion-dollar question in paleoanthropology. And the finding here, when they applied their ESPGS analysis to archaic genomes, is perhaps the most intellectually challenging, maybe even controversial result in the entire study.
Okay, I'm braced. What did they do?
They managed to get high-quality, high-coverage genome sequences from 4 archaic individuals. 3 Neanderthals, one from the Altai Mountains, one from Chukorsky Cave, one from Vindija Cave, and one Denisovan, also from the Altai region.
High coverage is key, means reliable data.
Absolutely. And then they ran their evolution-stratified polygenic score analysis on these ancient genomes. Crucially, they calculated 2 different types of polygenic scores for each archaic individual, comparing them to the modern European reference population from the epsilon study.
2 scores. Which ones?
First, they calculated the HAQER CPPGS, that's the annotation score we discussed, derived only from variants within the HAQER regions, acting as their proxy for genetic predisposition towards language-related abilities based on factors F1, F3. Second, they calculated the background CPPGS derived from variants outside the HAQ, which acts more like a general measure of overall cognitive predisposition or maybe correlates of nonverbal IQ based on the rest of the genome's contribution to F1, F3.
Okay, so a HQR language score and a general background cognition score. What was the great genetic contrast they discovered when they looked at our ancient relatives?
It was a massive, striking genetic dichotomy. The archaic humans, Neanderthals, and the Denisovan consistently showed significantly elevated scores on the HQR CPPGS. Their average Z-score was around plus 0.91, almost a full standard deviation above the mean of the modern European reference population for this language predisposition score.
Elevated. Higher scores for language predisposition in Neanderthal.
Yes. But simultaneously, and here's the contrast, their scores on the background CPPGS were substantially reduced. They showed a mean Z-score of around minus 1.45, nearly 1.5 standard deviations below the modern reference mean for the general cognition score.
Whoa. Okay, that is genuinely counterintuitive. Let me make sure I understand this. We have traditionally viewed the emergence of complex, perhaps syntactic language as the ultimate defining trait that separated Homo sapiens from archaic groups like Neanderthals. That was our special thing. Yet these results suggest the specific genetic markers linked to language specialization via HAQRs are scoring higher in our ancient cousins, while the genetic markers linked to more general cognitive capacity coming from the rest of the genome are scoring lower in them.
That is exactly what the polygenic score analysis indicates. It's a pattern that really forces a reevaluation of the timeline and the nature of cognitive evolution in the hominin lineage.
How do the researchers, and how should we, interpret that apparent paradox?
The interpretation put forward is that Neanderthals and Denisovans likely possessed an already elevated genetic predisposition specifically for the language-related abilities that are linked to the HAQERs. Perhaps the foundational aspects of grammar, vocabulary capacity, the specialized regulatory architecture for language. This core capacity, driven by that early, rapid evolution of HAQERs before the split, was likely a shared hominin trait present in our common ancestor.
Okay, so the language hardware might have been largely shared.
Potentially, yes, at least this HAQER-related component. However, their genetic scores for the more general cognitive factors, the ones linked to the vast, more slowly evolving background genome outside of HAQERs, were demonstrably lower than the modern human average.
So maybe they had the specialized language chip, but perhaps less overall processing power or RAM to run it as effectively, metaphorically speaking.
That's one way to conceptualize it, yes. It suggests that the key cognitive differences that ultimately distinguish anatomically modern humans might not have resided solely, or perhaps even primarily, in the evolution of these specific HAQR-based language circuits, since those seem to have been established earlier and were shared.
If this specialized regulatory hardware for language, these HAQR enhancers, was already present and perhaps even slightly boosted in Neanderthals compared to modern references, what does that imply about their potential for complex communication? Does it mean Neanderthals could have had sophisticated language?
It certainly pushes the needle in that direction, genetically speaking. It suggests their biological capacity for handling intricate grammar, syntax, maybe large vocabularies, might have been considerably greater than traditionally assumed based on older, simpler models of human uniqueness.
Does this align with any archaeological evidence?
It does align, albeit controversially sometimes, with the growing body of archaeological evidence suggesting Neanderthals exhibited complex behaviors. Things like sophisticated tool manufacturing, the Mosterian toolkit, potential use of pigments, possible burial practices, maybe even personal ornaments. These behaviors hint at symbolic thought, planning depth, and potentially complex communication needed to transmit those skills.
So the genetics might provide a biological underpinning for those debated archaeological finds.
It provides strong support for the biological potential being present, yes. Genetically, this data supports the idea that the core regulatory machinery linked to language factors F1, F3 via HAQRs was established early and shared. Therefore, the defining cognitive leap, the thing that really separated modern humans perhaps, might not be solely in the presence or absence of these specialized language enhancers.
But maybe in subsequent, perhaps more subtle, changes in the background genome. Changes that enhance things like working memory capacity, cognitive flexibility, processing speed, or the ability to integrate information from different brain modules more efficiently.
Exactly. It potentially flips the script. Instead of the defining feature being the sudden invention of language genes in Homo sapiens, maybe the key evolutionary action that distinguished us happened later, optimizing the broader cognitive platform that supports and utilizes language, things like enhanced memory, more complex abstract thought, better social cognition, perhaps.
So the HAQRs laid the foundation early, shared by all later hominins, but modern humans maybe got a CPU upgrade later on from the background genome.
That's a plausible narrative based on this genetic data. Importantly, they also found that the actual HAQR sequences themselves and their inferred enhancer function, based on the mouse assays where Neanderthal Denisovan sequences were sometimes used, generally fell well within the range of variations seen in modern humans today. This reinforces the idea that the rapid adaptive phase of HAQR evolution largely finished before the Neanderthal-Denisovan split, and the core functionality was conserved. The capacity, or at least the regulatory foundations for it, appears to have been a shared inheritance. The evolutionary changes that truly separated us likely occurred after the main HAQR burst and possibly involved many subtle changes across the vast, slower evolving parts of the genome, perhaps fine-tuning how language information was processed or integrated.
Okay, so these HERs, these rapidly evolved hominin-specific enhancers, seem to have given us, and possibly our archaic cousins, an incredible adaptive advantage driving specialized neurodevelopment, potentially setting the stage for advanced language capability in all later hominins. A huge benefit. But we foreshadowed this earlier. Evolutionary speed, especially this kind of rapid, specialized innovation, rarely seems to come without some kind of cost or trade-off. What price did this burst of evolution possibly exact on our modern human lineage?
Right, the no free lunch principle in evolution. The potential cost, as suggested by this research, appears to be an increased susceptibility to certain severe human-specific psychiatric and neurological disorders.
This brings us back full circle to that initial finding in the epsilon cohort, right? Where the language factors F1, F2, and F3, the ones genetically linked to episoders, were already found to be intertwined with measures of mental health and behavior even in childhood.
Exactly, that was the first hint. The researchers then wanted to investigate, is this link direct? Is it just a correlation because language ability and mental health are complex traits influenced by many shared genes? Or is there something specific about the HAQER regions themselves that carries this vulnerability?
So how did they try to trace that potential vulnerability directly back to the HAQR sequences themselves?
They leverage the power of the GWAS catalog. This is a huge, publicly available database that compiles results from thousands of genome-wide association studies, which have identified genetic variants, SNPs, across the genome associated with hundreds of different human traits and diseases.
Okay, so comparing the HAQR locations to known disease locations.
Essentially, yes. They performed an enrichment analysis. They systematically overlaid the genomic coordinates of the 1,581 HAQR regions with the locations of all the known disease-associated SNPs listed in the GWAS catalog. They asked, do HAQRs contain, or lie significantly closer than expected by chance to, genetic variants that have already been robustly linked to human diseases in large population studies?
And was there a significant overlap? Did HAQRs pop up near disease variants more often than random chance would predict?
Yes, the overlap was highly statistically significant. HAQERs showed a substantial enrichment for proximity to GOAS-identified disease-associated variants.
Okay, so they are near bad neighborhoods, genetically speaking. But was the pattern random, linking to all sorts of diseases? Or did it zero in on specific types of conditions?
That's the crucial part. The pattern was not random. The enrichment wasn't for all diseases equally. It specifically zeroed in on variants associated with certain neurological and most prominently psychiatric conditions.
Which specific disorders showed the strongest enrichment signals linked to these rapidly evolved HAQR regions?
The conditions showing the most significant enrichment for having associated genetic variants located within or near HAQRs included some of the most challenging aspects of modern neurological and mental health. The top hits were unipolar depression, schizophrenia, and bipolar disorder.
Wow. Depression, schizophrenia, bipolar disorder. Those are major psychiatric conditions with huge impacts on human well-being, and they seem to have genetic roots intertwined with these regions that potentially facilitated language evolution.
Absolutely.
Well, it's a powerful and, frankly, quite sobering implication.
It really is. It suggests that the very same evolutionary process, the rapid selection that shaped these HAQ enhancers and perhaps contributed to our unique cognitive abilities like language, might have simultaneously, as a byproduct, generated novel genetic susceptibilities for these debilitating conditions.
How does this proposed adaptive trade-off work at the genetic or mechanistic level? Why would evolving these specialized enhancers make us more susceptible to these specific disorders?
It likely comes back to that concept of pleiotropy, or rather the lack of it viewed from a vulnerability standpoint. Remember how we discussed HAQs potentially evolving de novo from previously neutral sequence to become highly specialized regulators, bypassing the constraints on older multifunctional genes?
Right, they evolved to do one specific job, like activating a gene only in prenatal radioglia.
Exactly. Because they are relatively new, evolutionarily and highly specialized, they are observed to be under limited pleiotropic constraint themselves. They likely perform a very specific regulatory function, perhaps in a narrow developmental window or cell type.
So, unlike older genes with many jobs, they lack that built-in redundancy or broader functional integration.
That seems to be the idea. The hypothesis proposed here is that when subsequent mutations, maybe common variants, maybe rare ones, occur within these new, highly specialized HAQR regulatory regions, the functional consequences might be sharp and specific, rather than broadly disruptive.
Meaning, a mutation in a HAQR doesn't cause catastrophic failure leading to, say, fetal mortality, because it only affects one narrow function.
Potentially, yes. Instead of causing broad developmental collapse, mutations that slightly disrupt the fine-tuned function of a HAQER might lead to more specific, perhaps subtle alterations in brain development or function that manifest later in life as these complex, human-unique neuropsychiatric disorders, conditions like schizophrenia or bipolar disorder, where the specific regulatory function controlled by that HAQER is subtly broken or miscalibrated.
So evolution, in its drive for speed and specialization via HAQERs, bypassed the safety net of older multifunctional regulatory systems, but in doing so, it inadvertently created these very narrow, perhaps more fragile genetic targets. A mutation there has a very specific detrimental outcome related to higher cognition.
Precisely. It's presented as a potential cost of that specialization. Rapid evolution allows for dramatic functional innovation, unlocking new capabilities like complex language. But it can also generate new genetic weaknesses, new points of failure that simply didn't exist in our more ancient ancestors whose genomes lacked these highly specialized elements. The rapid divergence created a fantastic new capacity, but that very specialized capacity, when slightly misregulated by subsequent mutation, becomes a potential source of profound human suffering.
It's a stark example of an adaptive trade-off, seemingly inscribed deep within our genome, linking the evolution of our greatest cognitive strength to some of our most challenging vulnerabilities.
It appears that way, yes. A complex legacy of our rapid evolutionary journey. Hashtag outro.
Wow, this deep dive has genuinely reframed, at least for me, the evolutionary story of human cognition. We've journeyed quite a distance today. We saw the necessary shift away from that older, simpler idea of a single language gene like FOXP2.
Yeah, the story got much more complex, focusing on the regulatory genome.
And that led us to the discovery of HAQERs, these 1,581 specific non-coding regions defined by their incredible speed of evolution on the human lineage.
Forged, it seems, by an early, powerful burst of positive selection, happening before the human Neanderthal split.
And crucially, we learned how they function, acting as specialized hominin-specific enhancers, turning genes on or off, particularly during that critical window of prenatal neurodevelopment.
And doing so in really foundational brain cell types, like the radial glia, potentially shaping the very structure of the developing cortex.
But then we confronted the potential cost. We connected the dots from that ancient evolutionary speed and specialization
To a modern vulnerability, demonstrating how this adaptive leap, the creation of HAQERs, seems to have also generated a clear genetic susceptibility and enrichment for major psychiatric disorders in humans, including depression, schizophrenia, and bipolar disorder.
So the evolution of perhaps our most unique cognitive capacity appears tightly bound to an inherent, definable evolutionary cost.
It certainly looks that way based on this evidence, a profound evolutionary trade-off.
But I have to say, I keep coming back to that really stark contrast revealed in the archaic human genetic data back in Section 4.
Yes, the Neanderthal and Denisovan polygenic scores.
Exactly. The finding that they scored high on the HAQER-based language predisposition scale, potentially even higher than modern reference populations, yet scored low on the background scale related to general cognition, that finding really sticks with you.
It absolutely does. And it raises a really powerful, maybe even provocative question for you, the listener, to ponder as you think about this material further.
What's that question?
Well, if the crucial regulatory hardware potentially underpinning complex language, these HAQER enhancers, was largely established, functional, and seemingly shared with our ancient cousins like Neanderthals hundreds of thousands of years ago, what does that truly imply about the timeline of language emergence? Could sophisticated communication be an incredibly ancient hominin trait? And if that core language capacity, or at least its genetic foundation via HAQERs, was indeed shared, where then should we really focus our ongoing genetic search for the cognitive traits that ultimately did define and perhaps separate Homo sapiens from Neanderthals and Denisovans? Maybe the key difference, the final cognitive leap that made us uniquely human in the way we are today, wasn't primarily located in the further evolution of these specialized, rapidly evolved language regulators.
But perhaps instead it lies hidden within the subtle, slower-moving evolution of that vast background genome.
Exactly. Perhaps the critical changes involved genes governing things like working memory capacity, cognitive flexibility, information processing speed, social learning, or the intricate integration of all these diverse cognitive functions. Maybe it was that quieter, perhaps less dramatic evolution across the rest of the genome, not the flashy burst of the HAQERs, that ultimately